So today I thought I would tell you a little bit about the kind of research that we do in my lab and combining different kind of genomics data for the purpose of really understanding the relationship between genotype, the context that that genotype operates in, and various kinds of human traits and phenotype eventually. But what I was told is that this is a very diverse group of audience coming from computer science, biology, and medicine. So I really hope to be able to speak to as many of you as possible. But I also realize there's a lot of jargon involved in biology and genomics. So if there's something not clear, please raise your hand and I'll try to explain. But for me to understand the pace, can you raise your hand if you're coming from computer science? Well, and then what about biology and medicine? I guess the rest? Okay, cool. Okay, great. This is exciting. So to start, this is kind of a very high-level motivation of the kind of work we do. So, you know, as human, we're all made up of trillions of cells of different types. So in brain, we have neurons and glia. In our blood, we have different kinds of immune cells. And then our health and disease is really a manifestation of health and disease of our cells, which are governed by two factors: the genome that each cell contains and the environment that that genome is exposed to. So of course, the genome of each cell in a given individual is exactly the same, and that doesn't vary across our lifespan, unless we're talking about cancer, but I'm talking about general healthy human development. But what does change is the context of that genome in different cell type or based on the exposures that we have as individuals. So very broadly speaking, an overarching goal in modern human genomics has been to really relate our genome to the environment that it operates in and to cellular phenotypes and diseases that we can observe. And this is, broadly speaking, what I'm interested in. But what's also been really exciting in the last 10 or 20 years is that there have been major breakthroughs in measurement technologies that now we have at our hands. So three types of data that are readily available in large human studies are genotyping data, where we can generate basically DNA sequences for a large number of individuals to try to understand their genetic differences. But on top of that, we also have assays that allow us to quantify gene expression and epigenomic data. So broadly speaking, of course, sequencing data, genotype data, is static. So you only have to measure it once for a given person, and it doesn't really matter which tissue you took. Generally speaking, the genome is the same across all their tissues and cell types. But what does vary is gene expression and epigenetic data. So we're taking snapshot of these dynamical systems to try to understand which genes are expressed in which tissues and what time. And broadly speaking, the hypothesis that motivates most of my work is that by combining these static measurement with dynamic measurement, if taken from the right time, we can try to understand how a dynamic system responds to its environment as a function of its genome, as a function of its exposures. So I'll just put in some terminology here just to make sure for those of you that are new to genomics. So in genotyping assays where we're measuring genotype data, we're really measuring one SNP or one single nucleotide at a time and trying to understand which alleles are present in which individuals. Gene expression data and epigenomic data, we're measuring in gene expression level of genes, and we have 20,000 or so expressed genes that we can typically measure. And epigenetic data, we're measuring something in relation to regulation of a gene, which is, again, dynamic, and I'll come back to this later. So in this talk, I'm going to tell you about two projects along the themes of relating genome to phenotype. The first part is very statistical, and here the idea is that how do we combine genetic variation across a large number of individuals to understand the function of genetic variation in the context of different environmental exposures or different cell types. And so that, as you'll see, that formulation of that is very statistical in nature. And the second part is much more better formulated as a machine learning task, where what we're trying to do is try to take DNA sequences, in particular non-coding regions, use machine learning techniques like convolutional neural networks to try to predict the activity patterns of those sequences in different contexts and different cell types. So the motivation for the first part is pretty obvious, and that goes back to genome-wide association studies. So one major modern way to figure out what genetic factors are associated with which disease is to perform a genome-wide association study, or so-called GWAS study. And what you do in these kind of studies is that you genotype individuals, and you typically have two groups, your individuals with a disease of interest and your healthy group, and then you scan across the genome to try to find SNPs that have differences in allele prevalence in cases and controls. So numerically speaking, what you do is you represent all these sequencing data in terms of number of minor alleles that was present at each SNP. So you get a giant matrix of this where you have tens of thousands of individuals and millions of SNPs here. And then you can start doing statistical analysis. So the standard thing that people do is use linear regression model or logistic regression model, depending on your outcome, to ask, is my phenotype associated with each individual SNPs? So through this exercise, for a given disease, you can identify a list of SNPs that are associated with that disease. So what we've done, what we as a community we've done over the last 10 years is a lot of GWAS study, and through these, several conclusions have become really obvious. So one is that for common diseases, so these are diseases that are very prevalent in the population, so talking about things like schizophrenia, cardiovascular disease, diabetes, you often have very polygenic genetic architecture. And what that means is that there is thousands to tens of thousands of SNPs that are associated with the disease, each with a small effect size. So this has a real consequence in terms of what we can do with this data. And the consequence is that we generally don't have good predictive power. So given someone's genotype, we can't really predict well whether or not they have the disease or not, because first of all, their disease is not explained totally by genetic factors. And second, each individual SNPs is explaining very that are uneven together. The ones that are associated don't give us enough predictive power, typically for most diseases, for most common diseases. But what this observation motivates is not just a pure prediction of disease based on genetics, but the idea that, okay, if we can't do prediction, maybe we can interpret mechanisms to understand what are the genetic factors and processes underlying the disease. And with that, we can go very far, because if we understand that, then of course we can design better treatments, right? And there are many common diseases we don't even have a single treatment for. So by understanding the genetic mechanisms, we can design better drugs. And just to give you a sense of kind of the number of GWAS studies that have gone on, I wanted to kind of summarize quickly this GWAS catalog. So GWAS catalog is an online database that basically collects the results of all sorts of genome-wide association studies that have been performed. So any kind of major GWAS study that was published, the results are usually dumped in this GWAS catalog. And if you go to this catalog, what you'll see is a map like this, where each line here is a chromosome. And along each chromosome, you have these points color-coded, which are basically SNPs that are associated with different diseases. So each SNP is a different, or sorry, each disease is a different color here. And you can say for a particular disease, where are all the SNPs associated with that disease located? There's a lot of data that we can now try to analyze to try to understand what are the processes behind each disease. So this is the state of GWAS catalog as of 2009. But if you go and look at GWAS catalog today, you'll see it's really exploded. So in terms of the number of SNPs associated with each disease, we really know a lot about which SNPs are associated typically with which disease. And you get a picture like this. Basically, I would say on the order of hundreds of thousands of SNPs associated with some human trait or some human disease. So there's a lot that we can learn from these associations, but the first thing that we need to figure out to do is how to assign SNPs to biological mechanisms. And this is hard because, as it turns out, most of these SNPs that are associated with any disease, they tend to be in the non-coding part of the human genome. So human genome, you can think of it part that are coding for genes and part of it that are so-called non-coding. So these are parts that are regulating genes but not genes themselves. And because most of these SNPs fall in these non-coding region, we generally have no idea what they do. So the next challenge after doing all these GWAS studies is to bring interpretation for all of these results. So one common approach one can take is to try to associate these non-coding SNPs to some sort of regulatory activity by linking them to genes that they regulate. And if we can do this, then we can already understand a lot because we know a lot about genes. So if we knew, for example, SNP1 is associated with schizophrenia and it affects gene B, then we automatically have a lot of resources at our disposal to try to figure out what that process might look like. So the motivation here is that we can take these SNPs that fall in non-coding regions and try to figure out which genes they're affecting. And this is common kind of strategy that's taken. And this formalized into EQTL studies, or expression quantitative trait loci studies. So in EQTL studies, what you do is you have your genotype data for a large number of individuals, and you get your gene expression data for also the same individuals. So then you can start correlating variation at the genetic level with variation at gene expression across individuals. And through this correlation process, you can identify which genes' expression are affected by variation at the genetic level of which SNPs. So one piece of terminology is an EQTL. So an EQTL is basically a pairwise relationship between the genotype of a SNP and expression level of a particular gene. So when I mention EQTL, we're talking about a pairwise relationship. And then obviously the goal of these EQTL studies is to try to find these pairwise relationships that can allow us to map SNPs to genes. So from a statistical point of view, this is also actually pretty simple. We just need the right data to do this. So what you can do is you model gene expression level as a function of typically the genotype of one SNP at a time and a bunch of other covariates that you can adjust for. And through this simple statistical model, you can figure out which associations are significant by analyzing the coefficient. There's two tricks to these EQTL studies. The first is these are actually computationally very burdensome because if we were to consider all pairs of SNPs and genes, we would be talking about trillions of pairs, or billions of pairs at least, because there are millions of SNPs and there's tens of thousands of genes. So we don't want to do this for all pairs. If we do that computationally, that would be difficult. But more so statistically, we would have a huge multiple testing problem. So to trick around that is using biological common knowledge, which are SNPs are likely to affect the expression of a gene that's nearby to them. If they're very far from that gene, it's very unlikely to affect the expression, at least in a way that we can detect it based on this data. So by encoding that information, one can either do very restricted search to, for example, only look at pairs of SNPs and genes that are nearby, or do something more fancy in terms of have some sort of regularization that's dependent on the distance between the SNP and gene. So that's one trick. The other trick is, it turns out that adjusting for covariates in your association analysis is really important because the relationship between genotype and gene expression is there, but it's weak because gene expression is also affected by multiple other factors. For example, the age of an individual, the sex of the individual, maybe what medication they're taking, and those have no relation to the relationship between genetics and gene expression. So if you had a bunch of covariates that you can adjust the gene expression for to explain some of its variability, then the leftover variability is better explained by the genetics. So over the last few years, there's been a lot of these kind of EQTL studies looking for relationship between SNPs and gene expression. And through finding these relationships, these studies have been able to explain the mechanism for a lot of different disease-associated SNPs. So I'm listing some that I was fortunate enough to be involved in earlier in my career. So for example, one of the first studies in this direction was the GGN study that looked at genotype and gene expression data for about 1,000 individuals to try to derive these links between SNP genetic variation and gene expression data. But ever since then, there's been multiple large studies, including the infer project, and the GTEx project, very famous, looking at gene expression from 1,000 individuals across 50 different tissues. So one thing that I'll point out is that maybe you noticed when I was talking about the GWAS, I said you need tens of thousands of individuals to find anything. In EQTL studies, you need less, you need fewer. If you have 400 or 500 individuals, you can already start finding a strong signal, and that's because, genotype is just directly upstream of gene expression. You're not looking at a very distant relationship. So these kind of a relationship you can find with just a few hundred individuals. So there's been 3 key observations from these EQTL studies that is going to motivate the project that I'm going to tell you about. So the first observation is that SNPs that are associated with disease are likely to be EQTL, it makes sense. That's what we were hoping for. So if a SNP is associated with disease, it's also going to affect gene expression of some gene. And this has been really fruitful in terms of figuring out their mechanism. The second is more interesting and unexpected. So I mentioned that gene expression is a dynamic trait, and it depends on which tissue you collected the data and which individuals you collected that data. So depending on the tissue that you collected the data, oftentimes you find these EQTLs that are only seen in that particular tissue type. So for instance, if you collect gene expression from brain, you might find SNP to gene associations that are only seen in brain data. And there could be a simple explanation for that. For instance, genes that are only expressed in brain, they're not expressed in blood. If you use blood data, you will never find the association between genetic variation of SNPs that are regulating those brain-specific gene in any other kind of tissue. So this makes a lot of sense, but it turns out that there are these tissue-specific EQTLs that you can find reproducibly in just one tissue or one cell type, but these tend to be especially important for interpreting disease SNPs. In that, for instance, in GWAS for schizophrenia, a lot of the SNPs that are associated with schizophrenia are EQTLs when you look at brain gene expression data, and you don't really see them if you use other tissues. And again, this makes sense, but this has been a little bit more unexpected that this link is so strong in a way that people expect. And the third, and the one that is motivating directly what I'm going to tell you about, is this kind of large analysis that the GTEx consortium did when they derived EQTLs from 50 different tissues. So what they did was take all the EQTLs that you can derive when you consider this huge amount of data and ask how many of the disease hits in GWAS catalog overlap any of those EQTLs. And if you do that, they reported something like 50%. So what this means is that there's 50% of SNPs associated with human disease that are not acting as EQTLs the way we're doing the analysis, even if we consider all the tissues that we've measured. So the question is, what are these other 50% of SNPs doing, and can we try to figure out their mechanisms? So this kind of motivated our work to try to come up with some alternative hypotheses. So one potential hypothesis could be that maybe these SNPs are EQTLs, but in a context-specific manner. And the motivation of this goes back to a few studies that have reported things like there could be EQTLs where you see a relationship between SNP genotype and gene expression, but only in smoking individuals, or only individuals that are old, or only in females. So there could be these special links between genetic variation and gene expression that's contact or environment dependent. And biologically, this makes sense that this would happen, and it's very similar phenomenon to what I was talking about, tissue-specific EQTL. So there are relationships that's very context-dependent. And I've listed some of these studies that have reported so-called context-dependent EQTLs. So from a statistical point of view, finding such relationships are actually not that hard, given that we already have a linear regression model that relates genotype to gene expression. So what we would need to do is expand our linear model to include an interaction term. Let's say we have expression level of a particular gene. We model it as a function of genotype. Now we include an environment. For example, this could be smoking. And then we have an interaction term between environment and genotype. And what we're looking for is the joint impact of environment and genotype that's significantly predicting gene expression level. So as an intuition for that, it could be that if you're a smoker, you're activating certain pathways that require regulation of certain genes, and you're not going to see the association between the SNP and the gene it regulates unless you expose the individual to smoking. So this is what we're looking for, this interaction term here, and that would allow us to detect these context-dependent EQTL depending on the environment we use to define our context. But now here so statistically speaking, this is not very challenging. You can obviously do this. You would have to pay a huge multiple testing burden depending on number of environment that you're using. Now you have to multiply that by all the other pairs of associations that you were testing. But that's still not the big problem. The big problem is that we actually don't have the ability to define high-resolution, detailed enough environments per individual to do this in a very detailed manner. So majority of studies that have the data have been collected, we really don't have that much metadata for individuals in this study. So at best, what we have is, what's the age of the individual, what's their gender, maybe if we're lucky, what medication they take. But that's all we pretty much know about individuals in this study. So, and even if you think about it, what does it mean to really at high resolution measure individuals' environment? That's not a really worked out concept yet. So the question is there some way to get at this question using tools from machine learning and statistics? And our idea was that why don't we try to infer environmental exposures directly from gene expression data. So that might sound like a crazy idea, but let me give you the intuition behind that. So it's well known that if you take gene expression data across a large number of individuals, you can do dimensionality reduction methods. You can apply dimensionality reduction methods like principal component analysis. If you then take your PCs or top principal components from the gene expression data and you correlate those with known factors, for example, batch effects or technical factors, or even things that are measured like gender, age, and maybe medication intake, you often see that your top principal components correlate strongly with some of these kind of factors. So that's what I'm showing you in this plot here. So this was gene expression data from a large sample called DGN, which has about 1,000 individuals. So I have done a PCA analysis where I have top 100 PCs here sorted by amount of variance they explain, and then correlate each of these PCs with a bunch of variables that we've measured on those individuals. So the top here are kind of technical factors that we're not that interested in, for example, which batch each sample was run on. But beyond that, we have things like sex, medication intake, whether or not person exercised, whether or not they ate something before the blood was drawn. And then the heat map shows the p-value for the strength of the correlation between these top PCs and each of these variables that we have access to. So a few note is that your top principal components often really strongly associate with technical factors, and this is well known, and it's actually a concept that has been implemented as part of normalization of gene expression data to get rid of your technical factor by exploiting the fact that top principal components typically correlate with those technical factors. But what's more interesting is that variables like sex, smoking, medication intake, as I mentioned, and whether or not a person ate before their blood draw actually correlate with some of these maybe later on PCs. So for instance, this sex is here. It's correlating with multiple PCs, some of them very strongly. Then we have smoking here. It's correlating with PC number 50 or something like that. We have our BP medication correlating with something here, and then 8 before blood draw is correlating with something here. So I guess the overall intuition behind that is that some sort of unsupervised analysis of gene expression data can get us some variables that typically correlate with exposures of different individuals. Now, here's a hypothesis. I could have one principal component here that maybe doesn't correlate with anything that I know, but maybe it's correlating with some sort of hidden or unmeasured environmental factors. So here comes the idea. So why don't we do this kind of factor analysis to come up with a large number of, I call them hidden factors or latent variables that kind of explain all the variability in gene expression we see across our individual and use those in this kind of G by E interaction analysis. And sorry, my equation here somehow got garbled, but that's supposed to be the interaction term. So this is basically what we propose is a two-step process. In step one, we apply some sort of factor analysis to identify latent variables or hidden factors from gene expression data itself. And in the second step, we just include those variables in this model to detect G by E effects. So one thing that I'm going to kind of sweep under the rug a little bit is that how do we actually extract these factors? So I mentioned PCA before, and it seems like, OK, that's a natural way to find factors in the data. But it turns out it actually works pretty crappy because it has this orthogonality constraint between PCs. And that tends to really destroy biological interpretation of PCs. And maybe you noticed that when I was showing this heat map before, for example, sex, which is one variable, it correlates with multiple PCs. So it really like kind of is diluting the signal in some way. So instead of using PCA, what we use is a method that was recently published called Plier, which is basically a constraint factor analysis with the added component that each principal or each loading of our loading vector is constrained to be representing gene pathways, a set of gene pathways or gene sets. So in a way, it's tying our loading to representation in gene sets and pathway space. And this is really nice because what it buys us is that for each of our PCs, now we know which pathway it represents, and that automatically gives us a lot of interpretability for that factor. We can think about, for example, a factor is activating nicotine receptor, then we know probably that's associated with people smoking or not smoking in our data set. So in application, what we did was apply this approach to two large gene expression data sets that came with genotype from the brain. So this is ROS-MAP study that has about 700 individuals, and the Common Mind study that has about 500 individuals. So we combined these two and actually did a lot of replication analysis that I'm not going to tell you about to say how, if you find factors in one data set, can we replicate it in a second data set? And there's actually really nice correspondence if we apply such constraint-based factor analysis that I mentioned, because it really tries to find biological signal rather than things that might be more technical. So with the combination of these two data sets, what we did was derive about 150 factors based on kind of cross-validation and figuring out optimizing the parameters of the model, and then use these 150 hidden factors to detect and these interaction EQTLs, which I'm going to call G by E EQTLs, which stand for gene by environment interaction EQTLs that's been called before. So what did we find? So this is our hidden factors here, and I have the number of significant G by E effect for each factors. In total, I would say we found something about 500 G by E EQTLs, which actually, to me, it was a little bit disappointing. I was thinking that interaction effects should be explaining a lot more variance in the data, and hence maybe would be a lot more prevalent. But regardless, we did find some of these G by E effects that otherwise wouldn't have been detected. So by modeling these interaction effects, we can explain about 4 to 10% extra variance in gene expression. So that means sometimes they have a significant effect, these interaction variables. Here's just one example that was kind of interesting. So we actually dug down on a lot of these associations to try to figure out what can they tell us mechanistically about SNPs that are associated with different brain diseases. Here was, or brain-related diseases, I should mention. So here was one interesting example. There was this SNP here that's associated with childhood onset of retinal dystrophy. And really, the mechanism for the SNP was not really known because it was in the non-coding regions. So what we realized is that if you look at the interaction between the genotype of this SNP and one of our hidden factor, then there's a significant effect on gene expression of a gene called SPATA7, which makes a lot of sense for this disease because that gene is involved in retinal development. So here was one example where we didn't really know which gene the SNP was associated with, but by considering its interaction, we were able to identify it. So I'll summarize here because I want to move on to the second part that talks about more machine learning stuff. But just to give you a summary, so I think there's still, with these G by E QTLs, maybe we can find additional EQTLs that we didn't know before, but there's still a lot of room for finding SNPs that affect cellular processes through secondary mechanisms. And maybe some of that comes from different kinds of data that we can collect, or maybe some of it comes from just having larger data set, but that's kind of an ongoing topic of investigation that is really exciting. So second part, I'm going to go more on the deep learning side, but maybe I'll just pause here just to see if anyone has a question or any clarification that I can provide on the first part at least. No? OK. So I can, and maybe I'll just ask you how well I'm doing on time to figure out how slow or fast to go for this one. Do you know? I'm good? Is it halfway through? Or? Yeah, OK. OK, so the motivation of this goes back to this interesting dichotomy in biology where we have progenitor cell giving rise to very different cell types that they all contain the same genome. So how is it that we have cells arising from progenitor with very different functionality, yet all of them having the same genome? So for non-biologists, maybe this is obvious, but for biologists, this is obvious, but for non-biologists, this is always like a very confusing and interesting puzzle. So how does that happen? So the answer, at least the framework for thinking about this, has been known for a long time, which is, I call it differential interpretation of DNA. But really what this is that differential usage of different parts of DNA by different cell types. For instance, I have highlighted in color different parts of DNA. These could be regulatory elements that are used by different cell type depending on their color. And a computational analogy is that all the cells have the same code base, but they're using different libraries and combinations of those libraries. So we know at a very high level how this works, but we don't have the details worked out for any specific cell type, how what are the regions that are different, and what are embedded within these regions that allow the cell to do its function. So to give you a little bit more background, basically the idea is that if you have two different cell types, they're using different regions of DNA to code for regulatory elements that regulate downstream gene expression level that can then implement cell type specific gene expression programs. And then by design, if you think about all the regions that are differential in one cell versus the other, then there should be some sort of sequence composition differences between them. And these sequence composition differences between them allow for encoding of sequence preferences for transcription factors that can then recognize these differential sequence preferences, bind to those, and regulate gene expression downstream. So this is conceptually, it doesn't seem that hard, but what makes it hard is that the space of gene regulation is very complex and complicated. So even though we have 20,000 genes, we typically have hundreds of thousands of regulatory regions that could be used in combination in any given cell type to regulate gene expression. So what is known, not known, is what are the sequence motifs that are present in regions that are specific to each cell, and what combinations work together that can define a particular cell type. But what's been really exciting to push these kind of questions forward in the field has been assays, like ATAC-Seq assays, that allow us to go and say, for a given cell type, what are all possible regulatory regions that are active or are regulatory in that cell type. So I'll give you the idea of how ATAC-Seq works. So in an ATAC-Seq experiment, you're exploiting the fact that DNA is not linear, but is wrapped around nucleosome in any given cell type. So it turns out that depending on how tightly the DNA is wrapped, that correlates with whether or not it's open, and that correlates with whether or not it has some sort of regulatory potential. So we have these nucleosome that the DNA is wrapped around. Some regions are so-called nucleosome-free, so you can see long stretches that are not wrapped tightly. And it ends up that these regions that are not wrapped so tightly are very likely to be enhancers or regulatory elements that are regulating the expression of genes. And so in the space of cell type specific gene regulation, these become really important. So what you get as the output of an ATAC-Seq experiment is a signal track like this. So at each base in the genome for a given cell type or a given population of cells, you can say how nucleosome-free that region is, and that correlates with the height of the signal. So then you can take the signal track, which is like a base pair signal across the genome, to identify all positions that were open, so-called open, in that cell, and those positions correlate with regulatory element for that cell type. So now the technology has progressed so much that you can do this experiment in parallel on multiple cell types. So this was a paper from just 3 years ago where they looked at 20 different immune cells, but I'm just showing you a few of them here. So for each immune cell, you can go and construct these tracks across the entire genome and say, which regions are open in which cell type. And that really defines the space of all possible regulatory elements for each cell type. So it gives you a really nice data set to try to figure out these mechanism questions in terms of which mechanisms are specific to which cell types. So one terminology that I'll define is OCR, or open chromatin region. So an OCR is just basically a peak that we can detect with a peak calling algorithm. And for each OCR, we have a quantitative signal for each cell that we have done an experiment with. So you can see this is a signal for one OCR. So this one is very active in NK cells, CD8s maybe, and maybe nowhere else. So how do we go from these measures of openness or potential regulatory activity to actually understand what's behind, what's deriving cell type specificity? So one thing that we can do is a simple differential analysis. So you can take all the regions that are found in NK cells and all the regions that are found in, let's say, CD4 cells, and compare those sequences with each other to try to find motifs like this that are maybe abundant in peaks that you find in NK cells, but they're not present in CD4 cells. So that's kind of a standard intuition behind what you can do with this data. And of course, things are very complicated. It's not just one motif. Typically what happens is that for any given OCR, you have multiple motifs that are binding, so the code of regulatory space is pretty complicated, but this is what you want to get to figure out what motifs and combination of motifs are important for each OCR, and based on that, define kind of the space of regulation or space of interactions between these motifs that define each cell type. So one way to formulate that problem is in terms of a supervised machine learning task, which is where it fits this problem really well. So you can say I have all my sequences, and I know which cell type they're active in. Can I develop or train a machine learning model that can take sequence and predict activity in 2 different cells? So most naturally, this could be phrased, for example, as a binary classification task. And actually, this kind of formulation has been around ever since the beginning of computational biology as I know it. So there's, for example, in 2003, there was a paper by Christina Leslie and others developing string kernels for precisely this task, where you take your sequence as input and then you try to predict some sort of binary output, whether and where is that sequence active in. And based on that, you can try to figure out what's the substring embedded within this region that's predictive of that cell type specific activity. But so with this kind of formulation didn't work so well in the beginning years of computational biology. One was that we didn't have the right data, and second is that it requires feature engineering. We have to come up with a lot of kind of features that were traditionally like k-mer-based features. So the search space really exploits when you start looking at longer and longer k-mers. And the type of data that we're getting today is actually much more complicated and complex and potentially allow for more flexible and more modern methods. So this is an example of a data set that we've been working on that came out of the IMGen consortium. So what they did was for about 90 different immune cell types, these are well-defined populations of cells, and there's even more than 90, but they only profiled 90, that they all exist in adult mice and also adult human, but this experiment was for mice. So for each of these cell types or cell population, they generated an ATAC-Seq profile. So we can now compare any two cells and say what are the regions that are differentially accessible in one cell type and not the other. So this kind of data gives a lot of powerful basis for doing something much more complicated in terms of how you can model it. And for this, we decided to explore convolutional neural networks, which are really nice in this case because, first of all, you can naturally model multiple outputs. You don't have to constrain yourself to binary classification tasks. And second, they don't really require feature engineering, and they can figure out the motifs and the combination that are important really by themselves without that much human input. So of course, neural networks have been around since the 80s, and I think when I was an undergrad in Toronto, one of the first algorithms that we were exposed to were neural networks because of Jeff Hinton was teaching the undergrad machine learning course. But what's happened in the last 10 years or so is that we really have, first of all, large enough data set to do this well, and second, we have really good optimization algorithms that allow us to fit parameters of these really complicated models. So, so what we decided to do was try to see how well a convolutional neural network will work in this case. And what we have here is basically similar type of formulation that I showed you initially, but let me elaborate exactly how we do this. So what we're going to do is take sequences that underlie each peak, and by definition, we've constrained our algorithm to find peaks that are about 250 base pair long. So the input sequences are sequences of 250 base pair long. And then we formulate a multitask or multi-output model to predict the activity profile of that sequence across 90 different cell types. So for each given cell type, we know the ATAC-Seq intensity, and that's basically a vector of 90-dimensional real value output that we want to predict. And so to be more precise, so the data that goes into this, I mentioned, is from this ImGen project that through this, looking at all these 90 different cell type, we've come up with 500,000 OCRs, or short DNA segments, and for each one, we have an activity profile across all these 90 cell populations. This is like a bird's eye view of what the data look like. If you have like our OCRs, or these open regions on the rows, and the cell type activity profile on the columns, what you'll notice is that data is structured and very blocky. So some OCRs are only open in types of T cells, some are only open in myeloid cells, and so on. So the structure is not random, it's a very strong structure that you see in that data that makes sense. But what we've realized for the neural network to actually work well is how do you encode the loss function? Because the neural network will learn the relationship between your input variable and the output variable under virtue of trying to minimize the loss that you define for saying how well it's actually making its predictions. And in this case, maybe it seems natural because your values are real valued to use something like mean squared error. But from our experience, that was a terrible idea, which I don't quite understand, but I can show you the results later. But what we realized that if you use correlation loss, it actually works really well for recapitulating this kind of very blocky structure. So what's a correlation loss? Imagine that for a given OCR, this is your hypothetical ground truth, where that OCR is very active in macrophages, monocytes, and not so much in T cells, and maybe intermediate level in B cells. So if the model makes a prediction like this, it has high correlation with this ground truth. If it makes a prediction like this, it has very low correlation with the ground truth. So we basically measure the correlation between the model's prediction and the actual observed value as a form of loss to encourage the model to recapitulate this kind of fine variation. And correlation works really well because we actually have a lot of structure in the data. So what we really want is to figure out which cell type an OCR is high in and which cell type it's low in. We care about those fine differences. So we've trained this model on proper cross-validation. So we train on 90% of the data, and then we test our performance on our 10% test set. So here I'm just showing you a cherry-picked example. So this is example the prediction we make for one OCR, which is a 250 base pair DNA sequence. So the prediction is supposed to be a vector of length 90 for the 90 different cell types that we've measured, which are kind of arranged along the lineages. So this is ground truth for that OCR. So we know that it's high usually in a subset of T cells and low everywhere else. And this is the predictions that we make. And what's remarkable is that we can even recover these fine variation within the T lineage, which is really nice. So the model has learned something about motifs that are present in that OCR that allows it to say even which of the T cells this OCR is more active in terms of regulatory potential. The other thing that we've noticed is that if you look at coefficient of variation computed from the intensity of OCRs across these 90 cell type, there's a strong correlation between coefficient of variation and how well we can predict the output for that OCR. So the more variable a given OCR is, so if it's absent in some of the cells and really highly active in others that would have high variability, we can predict those well. But the OCRs that are kind of constitutively active, we can't actually predict them that well. And that's interesting because traditionally when people have done similar thing, it's been reported that they can predict constitutive active sites better than the variable sites because there is some low-level sequence composition that allows you to do that prediction. But I think we're seeing this trend because we're using correlation loss that really focuses the model to figure out patterns that are distinguished different cell types. So here's just, so I show you kind of a cherry-picked example here, but here's more representative. So these are kind of OCRs that are well predicted, and you can see they all have these blocky structures. So this is the actually predictions we make for these OCRs. But if we go and use, this is based on correlation loss model, but if we go and use MSC loss that I mentioned might be more intuitive, what you'll see is that we can predict OCRs that are more constitutive better. These are the ones that don't have so much variation from cell to cell. So the model is using the same data, but different loss function. Obviously, the model is learning something different. And I think that's really important, how we formulate the loss function to try to get the model to learn what we want it to learn. But OK, so I mentioned about we can make these nice predictions, but that's really not the goal because we actually know which sites are open and which cell type. The whole purpose of this exercise was to learn a good enough model that we can then go and interpret to learn about biology and mechanisms that it's using to make those predictions. So to give an analogy, so the area of interpreting convolutional net or deep learning models in general is very active and very exciting right now in machine learning. And we know from image analysis that if you look at a convolutional neural network that have been trained on, say, like images to predict cars, then different layers of the network are learning different kind of abstractions. So for instance, in layer 1, these neurons are learning about edges. If you train the model to predict cars in this case. Layer 2, maybe they're learning about higher level objects like cars and windows. And in the later layers, they're putting together these different levels of abstraction to make a coherent prediction about whether or not a car was present or not. Turns out that something very similar is going on in biology. So the first layers tend to learn these like short motifs that represent transcription factor binding sites. The later layer learn combination of these motifs that are working together at a particular OCR, and then final layers are combining these combinations to try to figure out which cell type each OCR is active in. And that's exactly what we want to get at. What are these motifs? What are the combinations? And how can that tell us about which combinations are needed for prediction of which cell types? So this is just bird's eye view of the result again. So, and by construction, and I won't give you a lot of details, but feel free to ask me afterwards. So by construction, in our first convolutional layer, we had about 300 neurons, or 300 filters as they're called. So each filter is detecting a short motif. What I'm showing you here is these are 300 dots for these 300 motifs that we've learned. On this axis, on the x-axis, I have the information content of that motif, which basically tells you how different that motif is from just background prevalence of each of the 4 nucleotides in the genome. So the more complicated motifs have higher information content. And on the y-axis, I'm showing you what we call predictive influence, which is basically if we remove that neuron from the model, how much loss do we incur? So it's a measure of how influential that motif is for making predictions in our model. So you see these motifs that we've learned, some of them have high influences, some of them have low influences, but what was actually really interesting and surprising to me is that if you go back, so we have 300 motifs, now we can take these 300 motifs and match it to what we know from biology, and we can do this by going through databases that people have developed for over 30 years of kind of biology and bioinformatics to define motifs for different transcription factors. So now I'm coloring these motifs by whether or not such a motif existed in a database or not, so annotated or not. We knew anything about the existence of that motif in a database like SysBP. And what you can see is that a lot of these influential motifs that we're finding are actually known biologically to be important motifs for transcription factors. And in some way you can say, well, what did you even learn? We already knew this, but my answer to that is that we were recapitulating 30 years of biology and bioinformatics in one experiment. The experiment is applying deep networks to powerful data, basically. But so this is interesting that we're finding a lot of these, but what you'll notice that a lot of the motifs on their own, they're not very, they don't have any, a lot of influence, but what do they have is the combination of them that are influential together, and that makes perfect sense biologically because there's a lot of redundancy in the system. So next thing we can do is, for each motif, we can figure out which cell type it's important in, and that's by taking out that motif from the model and observing the loss across our 90 different cell types. So we can say if you, for example, removed the ETS motif, which cell types are most affected, and that's kind of like in silico mutagenesis experiment that's kind of really cool. So now we have these 300 motifs here, and we have their profile of influence across our cell types, and a lot of these, again, are making sense with what we know from biology. So for example, the PAX5 motif that's known to be very important for B cells, if we remove it from the model, we see that we're hurting most in B cells that are here, and that kind of really jives with what we know biologically. And finally, the most exciting aspect is that now we can look at combinations. So which combination of motifs are typically working together in higher layers. And this is one way of trying to drive that. And I mentioned that the science of interpretation of deep learning network is really an active science. So this is just our heuristic way of doing that. And I'll be happy to give you details of how we do this. But so essentially with our process, we can remove 2 filters at a time or 2 neurons at a time and say, combination of 2 neurons, how does it affect our output, and how does that compare to removing 1 neuron? And based on that, we can come up with pairs. So we can find thousands of pairs that are co-influential together, and some of these, again, make a lot of sense with biology. But the last thing that I'll show you is kind of the ultimate test of generalizability of the model. Because every time you train a machine learning model, there's this fear of, did I overfit my data? Maybe my test performance is not really as good as I think, because there's, even if there was no cross-validation leakage, but maybe there's systematic artifacts in the data that make your performance look better than what it is. So the ultimate test is just to take this model and apply it to a setting that the model knows nothing about. And in our case, we got lucky. So all of this MGen data was from mouse. So we got lucky that was a similar kind of data available from human that the model had never seen before. So there was a paper by one of our collaborators where look at ATAC-Seq data for about 23 different immune cell types in human, most of which overlap with some of the 90 cell types that we had in mouse. So we can now go and directly ask, if I take my model that's entirely seen mouse sequences, how well does it do in predicting activity profile of these human sequences? And that was actually super encouraging. So what we saw is that, so this is the performance of the model, again, measuring it in terms of correlation between the ground truth and models prediction across the 20 or so, I think, OCR cell types that were shared by mouse and human. So you can say the average correlation is 0.55, but there are many OCRs that were predicting really well. So we know a sequence, if we see a sequence, we know which cell type it's going to be active in. And then you can compare that with shuffled sequences. So if we were to take the human sequences, shuffle them, the prediction really goes away. So there's nothing there, but on the real data, there's structure that we are capturing using mouse data alone. So with that, I will conclude by saying that I'm super excited about this direction. I think it was one of those things that there was a lot of hype about deep neural network and what can they do. And I said, oh, I'll believe it when I see it. But now I've seen it, and I'm convinced that for this kind of problem, going from sequence to some sort of functional output, they work really, really well. But I'll end with saying that extraction of what the model has learned is really an ongoing area of research that needs a lot more thought and a lot more smart people to try to figure that out. And then finally, just thanks to the collaborators. So I mentioned the MGen project, which was all these nice ATAC-Seq data from 9 different immune cell types. That was all enabled by this MGen consortium that's led by Christophe Benoit. That's been a really key collaborator on this project with us. The first part, we worked very closely with ROSMAP consortium and people, including Phil Diager. And then the work that you saw was from my student, Sasha, on the deep learning project, and Bernard on the GBAI EQTL project. And thank you so much for your attention, and happy to take any questions.